Good afternoon, everyone. So today we have Dr. Wei Yang from National Institute of Diabetes and Digestive and Kidney Diseases (NIDDK). She's a NIH distinguished investigator. She studies DNA recombination, repair, and replication. Her lab is interested in VDJ recombination, mismatch repair, nucleotide excision repair, and translesion DNA synthesis. Just a little bit of background on Dr. Yang. She majored in biochemistry from Fudan University, Shanghai, and SUNY at Stony Brook. And then she did a PhD with Wayne Hendrickson in 1991 at Columbia University, and did a postdoc with Wayne Hendrickson, and then with Tom Stite from 1992 to 95 at Yale. And since 1995, she has been an investigator at NIDDK. Of course, Dr. Yang has won numerous awards, including the 2011 Dorothy Crowfoot Hodgkin Award, and she's also elected member of the American Association for the Advancement of Science and the National Academy of Sciences and the American Academy of Arts and Sciences. Today, she's going to talk about the formation of our adaptive immune system, from VDJ recombination to NHEJ. So let's together welcome Dr. Wei Yang.
Good afternoon. Thank you, CJ, for having me here. And I feel a little guilty. It's so beautiful outside, and I'm keeping you inside here. And I've been here before, but never in such beautiful spring days. So it's really nice. So my research program encompasses many different things, but today I'm going to focus on one topic: is how our adaptive immune system, T cell, B cell forms and form the antibody and antigen receptor to defend us from any invasions. So the project started, actually, my interest in the topic started when I was a graduate student, and I was taking class about how DNA, the specific sets of DNA recombination happening in human cell, but we were not able to do anything for good, like 20 years until like 2000, we were able to do things. Then cryo-EM revolution changed the landscape, and things get faster and more. So I'm going to give you an overview of from beginning to the end, so you don't have to follow everything. And I know not many people work on DNA, but at the end of the day, nucleic acid are similar, RNA, DNA are not that different. Some of the principle, hopefully, is going to be held. So after COVID, I feel like everyone feel like we are somewhat immunologists. We know how antibodies get induced, just vaccinate us. Somehow it's amazing. Every one of us are magician. We can create antibodies like here. This is an antibody showing green and can specifically recognize antigen. This particular slide is generated by Peter Kwong. It's like a GP120 of HIV recognized by specific antibody. And the antibody recognized the antigen uses this particular variable region showing yellow. And it's like a Y-shaped molecule. And so the ability for us to can create unlimited amount of antibody to fight against any of the antigen that's introduced, like vaccines, because we don't have the whole gene to every antibody chain have to be encoded by different genes. So shown here, the antibody is made of 2 light chains, the shorter ones, and the 2 long ones are the heavy chain. And the gray part never change, and not never change, they're very little change, have different classes. But the major change are those top region called the V segment, J segment on the light chain, and the heavy chain, the V, D, J segment. And those segments, each segment have variations by stochastically connect V to J and the V to D to J, and imprecise joining. Then we create this yellow region, that's the tip of the antibody. It can be varied, and we can make unlimited number of this variation. And upon exposure to the antigen, then specific interactors get selected and being committed to memory, and then refined, have somatic mutation to make it even more specific mutations. So the process is pretty complex. I give you an overview today. So each of those segments will be cut and recombined, so-called only active site-specific recombination active in human. And the defect in this specific recombination, which make our adaptive immune system, is not causing life and death. You know, baby can be born, children can grow, but they don't have the immunity to defend ourselves. So it's famous like a bubble baby, if like they known have genetic defects, so-called severe combined immune deficiency. They have to be used to be brought up in a confined environment, completely sterile. And it's very hard. And in recent years, since 2018, the gene therapy worked. If there's a monogene defect, and there are 4 different monogenes now successfully using lentivirus can be supplemented, introduced to those children, and they're fine now, but not all of them. So that's the story of VDJ recombination biologically. And in terms of the molecular mechanism, so each of those segments, V and the J, or V, D, and J, is flanked. Coding segments is on the left and the right. They're flanked by those triangle-specific sequence. So specific recombinase will recognize those sequence. And there's open circle and a closed circle, they're different, so that V and the V will not be joined, only V and the J, or D and the J will be joined. So recombinase is a recombination activating gene will recognize those specific sequence, make a pair. And then they have to cut the double strand, right, at the border of the triangle and the coding segment. And the chemistry is interesting because this is the biochemistry department. So it's a double strand cleavage carried out by one active site. The first is hydrolysis. They cut at the border of the recognition sequence and the coding segment, generate a 3' OH. So it's hydrolysis. After OH is generated, it does a nucleotide transfer. It's very much like a splicing, so RNA people can be happy, the same reaction. And the result is forming a hairpin, because the top strand OH attack the bottom strand. So the coding segment is flanked by hairpin. And the signal sequence for cleavage, the triangle is a blunt end. So this 2 cleavage, different type of cleavage is catalyzed by REG1, REG2 recombinase. And after hairpin generation to stitch together different segments, then there's a specific nuclease called Artemis, and that opens the hairpin, the coding region only. And then both the blunt end and the open hairpin will be joined together. One signal sequence will be form circles, so they won't invade any new sequence. So they can, during cell division, they disappear on the right side. And then the left side, the coding gene will be joined together. And the joining is imprecise. You can have deletion or addition of nucleotide. So the junction creates even more variation on the surface of antibody that face the antigen. So today what I'm going to show you is the whole process. It's a lot to digest. Some is going to be brief, some will be very more detailed. So starting from REG1 double strand cleavage to hairpin opening by Artemis and the final non-homologous end joining NHEJ. So to begin with, oops, it just went too fast. So we need 2 different triangles, as I mentioned. That's the V and the J segment, triangle difference. So V and the J will be paired up. And those have same 7, 9 base at each end of the triangle, but the intervening sequence is 12 versus 23 base pair. So they call the 12 and 23 recombination signal sequence, 12, 23 RSS. And only the 12 and the 23 will pair together, so make 2 double strand breakage. And the 12, 12 won't, 23, 23 cannot be paired. So the top is the DNA, then the protein is a REG1 and REG2. When I started in graduate school in 1985, 1,000 amino acid protein was too large. So people only think about divide and conquer. So each of the little domain structure gets solved. Ring domains, the E3 ubiquitinase, and then REG2 C-terminal, it's a PhD domain. But the whole protein was resistant to crystallography, and the protein expression was in it. issue. So when the protein was expressed in insect cell, it can reconstitute the recombination enzyme, but only 1% of purified protein have activity. And for structure biologists, if you have 1% of active protein, that's finished. We cannot crystallize or anything. So the real key to get it to work is we transfer the expression system from insect cell to mammalian cell. And that makes day and night difference. And it's not the post-rapid translation modification. It's purely because some protein chaperone is unique in mammalian cell and they can fold the protein properly. So Minson is the final one to get it to work. So he was able to grow crystal of the protein with DNA, and the protein with DNA. Initially, the DNA gets scrambled during crystallization. We couldn't see the DNA. And then he modified DNA to including not only the triangle RSS sequence, also the coding sequence. And then refined the system, so he got protein plus DNA. So the whole protein is a dimer of a heterodimer. The blue and the green are 2 copies of reg1, and the purple, magenta at top are 2 copies of reg2. And the protein alone looks like a Y shape, life mimic art. And this is the gene that make the antibody mimic antibody. And then once the DNA is in, remember the DNA is asymmetric. One's 12 base pair spacer, one's 23 base pair spacer, and the tip of the Y recognizes one of the dimers. So the protein becomes asymmetric when DNA is bound. So you will see a little bit of a movement. The tip we call the non-number binding region tilted over. So the orange DNA you see is having 23 base pair in the intervening 7-mer and 9-mer recognition. The recognition is end up in the active site, which is in the middle of the Y arm. And to help the DNA to bend almost 150 degrees is the very light blue color things. Good pointer. So that's called HMGB. It helped the DNA to bend. Each domain, the HMGB have 2 domains, and the one domain binds maximum 90 degrees. So it helps the DNA. Orange DNA is 23 RSS, is much more bent than the yellow DNA. And the complex once formed, the 2 active sites are linked through protein. So it will, each of the green and the blue right one will make double strand break. So they are coordinated. And so we have these 2 initial structures, and the later structure we couldn't make by crystallography. It wasn't very good. So we have to resort to thank you, for the cryo EM to come. So shown here is apoprotein, then protein plus 2 DNA, and 1 DNA is bent more than the other, and they all recognize the same sequence, 9-mer here, 7-mer here. The 23 and the 12 spaces in between. HMG helps the bending. Then we want to know the whole chemistry. So we have the protein DNA initial complex is DNA, but it's not ready for cleavage. To convert from complex of protein to DNA is able to be hydrolyzed. There's a big conformational change. I simplified the slide. Instead of showing you everything, I'm only showing you 1 DNA. So basically, there's 1 DNA need to be nicked. You will see the conformational change from initial protein DNA association to be able to nick. There's a huge unwinding, 180-degree rotation. DNA formed this we call zipper. So it's, instead of base pair, it's interdigiting, and DNA gets taller. So the cis-O bond can reach the active site. And then second step after DNA is nicked, actually both strands gets nicked around in V and the J segment, for example. Then after it's nicking, the second strand cleavage is hairpin formation. So remember, DNA is anti-parallel, so they will go to fitting to the same active site, so have to bend in order to mimic the same thing. So this is the second strand need to be cleaved is on this. So there's a huge conformational change, and the DNA is like bent more than 690 degrees, so that can form the hairpin. And I'll show you this again, so you won't miss it. So now it's putting the 2 together. That's the first DNA zipper formation. And you will see that both DNA strands unwind 180 degrees and grow taller because the cis-phosphate is here. It has to go to the active site, and DNA grow a lot taller. You'll see the repeat. And it's not protein DNA coordinate movement. They actually move in opposite direction. It's just the final conformation of this zipper DNA is stabilized by the protein conformation. There's no ATP involved. Everything is in the DNA sequence. The sequence bordering the cleavage site is CA, CA, GTG. And you reverse it, it's TG, TG, CAC. So it's the dinucleotide of TG and the CA. Today, I got a conversation with Sam, the pug, UG, it's the TG version. So those dinucleotide actually is utilized in biology a lot, in a lot of retrotransposon and contain CATG sequence because those purine pyrimidine adjacent to dinucleotide are particularly prone to deformation. And the Tata box is another example. And here we just have CAC is absolutely needed to form this zipper sequence. There's no particular protein DNA interaction to induce it, but CAC is prone for this conformation formation occurring, and the protein just capture that particular conformation for cleavage. And then for the second step of hairpin formation, there's a big scissor movement. So that you can see this is already cut. We feed the cryo-EM with the nicked one, ask for it to forming the second. So the second cysteine phosphate goes to the active site through this scissoring motion of the dimer. So the movement is mostly surrounding it, and it really need the docking at the end. That is like a rudder of the sailboat. So there's huge conformational change, and it's kind of really amazing how molecule can do all this. And then I will just focus you on that, how the DNA look like at the active site. So Xiaoming did a lot of work. So to get this, to stabilize those unstable structure, he has to mutate one residue to a basic site. So make the unwound zipper structure very more stable. So at the beginning, the cysteine phosphate is there, and active site is here. An active site composed of 3 carboxylate, and without the cysteine phosphate in the active site, the certain carboxylate is far away. So it's basically not cleavable. Nothing can be done until through the thermodynamic movement of the DNA, the cysteine phosphate gets into the active site. And coupled with the cysteine phosphate arriving, the active site form, and the trapped 2 magnesium ions got the nicking. Then you have the second movement, rearrange the active site to form the hairpin. So it's a lot of molecular acrobat, but that happens. And it doesn't need to happen very often. You know, each T cell and the B cell just need to go to a few times of those. Once you make the connection, that's done. So it's rather amazing. Hydrolysis and the nucleotide transfer reaction. And here, actually, I'm going to give you more detail. As a structure biologist, one is you kind of show how things are arranged, like I already show you here. But the second thing is, how does chemistry really happen, right? So, you know, the thing is, like I was speaking with students, sometimes like we didn't really engineer to see particular this one, but then other research projects are connected. So we studied DNA polymerase for a long time. DNA polymerase is an example of nucleotide transfer reaction. And I was trained in Tom Steit's lab, so he was looking at the molecular central dogma. So here's a template blue template, and the red chain is the primer. And it's textbook work because we just make into the cartoon that the polymerase have enormous specificity and depend on 2 magnesium for catalysis. If the wrong incoming nucleotide come in, it reject because the second metal ion couldn't bind. If the incoming nucleotide coming is correct, then second metal ion bind and align the 3' OH to do the nucleophilic attack of the incoming nucleotide and form this transition intermediate. Then the leaving group is pyrophosphate, and it's so-called acid-base catalysis, you know, deprotonating the 3' OH and then reprotonate the leaving group. So watch again. So that when the 2 metal ion is bound, supposedly deprotonate the 3' OH. Now I have a nucleophilic attack. This pentacovalent phosphate is the intermediate in every reaction, RNA synthesis, splicing, etc. And then we have a general base to deprotonate it. And this is based on many structural work, but it's never really the transition state, right? Because that's supposed to be transient. We're not supposed to see it. We just diagram it here, and the two metal ion were observed. And everyone is very familiar with two metal ion catalysis. And supposedly all enzyme catalyze the reaction but reduces transition state energy level. So that's the given. And for many, many decades, we just have the substrate state, product state, everything in between we imagine, and that's the proposal. Somehow enzyme stabilized the transition state we never saw. So, and while studying that, that's the pentacovalent intermediate. We just figure like, it's like a horse racing. The horse standing is very different from how a horse is running. And actually, this series of pictures was taken in 1878, way before there was a video camera. You cannot take continuous figure and you can stop. But even in 1878, by cleverly, Edward Maybridge, cleverly, he placed the camera along the racetrack and have a thin line when horse crossing the line will trigger the shutter. So he actually trapped those intermediate of how horse running by using the camera. And each camera takes one picture, but it's enough. You string them together, you see how the horse runs. And we clearly see when horse run, it's completely different when horse stand. So for enzyme, if we only look at this state, that doesn't tell us intermediate. And the reason he took this series of picture was because there are two people making a bet. One said when horse running, there's always one leg on the ground. And the other said, no, all four legs leave the ground. And with the picture, it's obvious, right? How horse runs. So we really wanted to see how the enzyme catalyzes a reaction, particularly in the middle state. And to see that, it means we need a crystallography, allow the reaction happen in the crystalline so we can trap the intermediate to photograph it. And so the first question is, does enzyme work in the crystal form? And the answer is absolutely yes. The very first enzyme ever crystallized is urease. And Jim Sumner actually crystallized the urease, and he got Nobel Prize. But I don't know how many people remember Jim Sumner won the Nobel Prize. But the fact is fact. And the people have shown since Jim Sumner that enzyme in the crystal form is reactive, including DNA polymerase. And Lorena Beasy shows you can always see the DNA grow in one of her crystal form, but all the structure he saw, she observed it was the product state. So we set out to do reaction in crystal and took advantage that the polymerase nuclease we study require magnesium. So we grew crystal in the presence either of calcium, limited amount of calcium, which does not support the catalysis. And so that we don't use any inhibitors. And then after crystals grown, I'm showing three people's face here and the name. They work on three different enzymes, one polymerase and the two different nuclease, but the same methodology works. And after crystal grow, and then we can wash away the calcium and then replace it with magnesium or manganese. So we prefer magnesium because it's more natural. The manganese help in crystallography because it's much more dense. So then we switch the protein crystals soaking in the magnesium manganese and sometimes using EGTA to deplete the calcium. And then once it's in the magnesium, depend on the starting 0 minute exposed to magnesium, we'll wait for 10 seconds, 20 seconds, 30 seconds, etc. time point, and then freeze the crystal in liquid nitrogen, and then at the liquid nitrogen temperature, everything stopped, so frozen. Once crystal frozen, we can take data, and that's the approach we took. And it worked out really beautifully, and we have multiple papers on this study of enzyme reactions. So I will show you two examples in actually movie. So in DNA polymerase, we were able to crystallize POL-eta, which is a translesion DNA polymerase. It's not for replication per se, it's a slow enzyme to begin with. So in the crystal, because molecular movement is limited, it's even more slower. So it's great, it's very slow enzyme. So we crystallized protein, Shown here, we're only showing the active site. Here's incoming nucleotide ATP, and everything is there. But this is the carboxylate, 3 carboxylates with 1 carbonyl oxygen. We're only giving each complex based on the total protein, 1 calcium. So even contaminant magnesium doesn't have a chance to have 2 magnesium in the active site. So the crystallization can take days and a week to occur, and we crystallize enzyme substrate together. No reaction. Then when crystal is grown, we actually even lower the pH for reducing the reactivity. Then we raise the pH and replace the calcium with magnesium. You see, when we only given 1 divalent cation, the 3' OH of the primer end here is not even pointing to the alpha phosphate. So there's no reaction possible. When we replace the green calcium with 2 magnesium, you see the alignment of 3' OH. Okay. So it's textbook, enzyme allowed substrate to be aligned. We actually studied this complex before. We were using the non-reactive incoming nucleotide, replaced the oxygen here with the magnesium, so no reaction. And the 2 structures superimposable. And we get this within 40 seconds of changing calcium to magnesium. And the surprise is, unlike what textbook said, when you have 2 magnesium ion, reaction happen. Actually, reaction doesn't happen. Everything's aligned, no reaction. And the only time reaction happen is when we increase the divalent cation concentration. Without the third magnesium ion, so this is just 2 metal ion, we just only observe the sampling of this regime. Brownian motion must happen. And when we increase the metal ion, a transient bound third metal ion come in. It's on the leaving group, helping to break the existing phosphodiester bond and allow the, so you watch it again. Alignment by 2 metal ion, no reaction happen. We saw water next to the 3' OH. And the only reaction happens when we increase the incoming, the magnesium concentration or manganese concentration. So a third transient metal ion comes in, help to breaking existing bond and force the new bond formation. And we believe third metal ion is the catalysis metal ion. That's the one dropped the barrier so that the reaction can go forward. And the reason we believe that is because we did a lot of experiment biochemically. I'm only going to show you one. So basically, in solution, you can titrate how much magnesium you need to increase the reaction rate to full reaction. So that's measuring essentially the catalytic metal ion, essential for reaction, and the binding constant. In this titration, you don't know how many metal ion bound, how many is needed. And in a separate reaction, I'm not showing you here as you can titrate. There are 2 different binding constants of metal ion in solution, but never one sees it. So when we do the in crystal reaction, we actually do the titration. We can actually measure each binding site, the binding constant. And the A and the B, 2 classic binding site, the binding affinity is much higher than what's needed for catalysis. And that's a similar binding constant. And no reaction happen when the 2 AB site is satisfied. And the C site, the third metal ion, which I show you, coming transiently, help the reaction to take off. The binding constant exactly matched bulk solution binding constant. And the third metal ion was observed in 3 other polymerase transiently during the reaction by Sam Wilson's group. And we also tested nuclease hydrolysis. So the reaction I just showed you was equal to nucleotide transfer reaction, but even in nuclease cleavage assay. So nuclease cleavage is much harder. This is RNase H, which cleave RNA when it's hybrid to DNA. So here's the RNA strand. And there was a lot of work by Nadine, like 3 years not having the thing to report. Eventually she has, it's published in NSMB. So different from polymerase, when she have 2 magnesium bound in the active site, the nucleophile water is not even aligned for catalysis. So inline reaction have to be there. So it's not aligned. And she had like 1.3 angstrom resolution, so she can see there's 2 potassium there to begin with. And then there's a certain magnesium took a long time to trap it because it comes in quickly, left even faster. So she had to do mutations to capture the certain metal ion. So when the certain metal ion, divalent cation come in, then you will see, I'm just slowing down here, the alignment, and then the cleavage happens. And we observe that certain metal ion in 2 different nucleases. And the reaction is actually quite complex. The reason I told you the certain metal ion is quickly gone is because after reaction, it's replaced by protein side chain, lysine, so it's done. So all we see structure is always this, unless we do something, mutate this to alanine, then we see the certain magnesium ion for hydrolysis. So we suspect this transient divalent cation coming is often with the nucleotide transfer reaction. Even for protease and lysozyme, we suspect potassium, etc., can come in and go in to help the reaction to go. So with this, and we give you so much detail, the next 2 parts I'm going to go relatively fast. So simpler. So the hairpin opening, after the coding end is formed, hairpin opening requires a specific enzyme called Artemis, and Artemis is the hunter goddess. And the protein is not so big, 692 amino acid. The nucleus catalytic domain is even smaller, only 360 residues. There's a lot of phosphorylation site, somehow regulate the function, but we cannot pinpoint exactly what the phosphorylation do. But what's interesting is this tiny little protein is the only enzyme in the human body that can open the hairpin in vivo. And if you don't have it, we don't have antibody antigen receptor. And what's interesting is this is Mike Lieber's work. They purify the protein, and this is the DNA substrate, just a hairpin, and the 5' end labeled. Artemis alone only have the exonuclease activity. So this is the full-length substrate, that's the 5' end label. And to get the hairpin opening activity, they need to add a protein that's called the DNA PK, which is a DNA-dependent protein kinase. So I need a big protein to help the little nucleus to open the hairpin. And when the structure of the nucleus itself was determined, and then you know it's why, because it doesn't bind to hairpin, it binds a very few base pair at the tip of double-strand DNA, and it's like a low occupancy, and the 5' end of the DNA is in the active site. Like it can recognize 2 or 3 base pair, not the hairpin. So what is DNA PK? DNA-dependent protein kinase PK is the largest protein we have studied. It has 3 subunits. The kinase subunit is 4,128 residue. And at the time, even the cloning wouldn't work. You just couldn't get the protein clone overexpressed. So we purified the protein from HeLa cell nuclei. And it's very abundant protein, thankfully. And so the protein was purified, and Q70, Q80, 70, 80 kilodalton protein, well known for binding DNA. And when the protein was purified by Joyce, and she also synthesized the DNA, and when she put together, she can make a complex. And tried to crystallize it. Crystal was diffracting very poorly, and Joyce had to take maternity leave. So the project was on shelf. And then cryo-EM come along, things happened. So I'm showing you the structure of Q70, Q80, the structure of the 2 proteins together bound to DNA was determined in 2001. But this portion of the C-terminal, the end of Q80 is not seeing the crystal structure. So Q70 is in pink and the Q80 is in magenta. And the extension of Q80 here, you can see it's dangling there is because that's where the recruitment of the DNA PKCS. So DNA PKCS is very large. We color it blue-green for the 2 heat repeats ring, and then the yellow is the fat domain, so-called, and the kinase domain is at very C end, orange. So you have the color of those 2 rings, blue and the green heat ring, and the head is made of fat in the kinase domain. We'll look at the bottom, how the DNA is bound. So DNA is bound by Q, but the very end of the DNA is surrounded by this green and the blue heat rings. And this is protection of DNA and open end by DNA PKCS. So it's a big protein complex. It serves 2 roles. One is helping Artemis to cleave the hairpin. The second, if the DNA end is open like here, it protects DNA so that the nucleus and other things don't get to it. So it can be stitched together during the G1 phase. So it's called the DNA-dependent protein kinase. Is when DNA is bound, you see large conformational change of the blue heat domain, and there's the head piece also moves. And the most important thing is when the DNA is bound by the 3 protein subunit, the kinase domain become activated. So before, there's this red helix blocked ATP binding, and then with DNA bound, it opens up so it can phosphorylate. So the interesting thing is that what is it? The kinase. This kinase belongs to PI3K family, belongs similar to mTOR, ATM, ATR. They all have this fat domain and a lot of heat repeat. And the structure of ATM, ATR, mTOR, they're still missing like the substrate, exactly how it bound to the kinase. And DNA PKCS become a good subject to study because it kinase phosphorylating itself. So it's a large protein and it has a very preferred kinase phosphorylation site, a cluster of threonine or serine here. And we call it, they were named ABCD cluster, the very first patch identified. This phosphorylation cluster is very interesting. If mouse without DNA PKCS at all, mouse is actually born. They just couldn't make VDJ recombination, so they are immunodeficient, but they are born okay. But if you kill the kinase activity or remove the serine threonine, make them alanine, cannot be phosphorylated, now the mouse is embryonic lethal, so they cannot even be born. And the reason is that this DNA PK complex 3 subunit protein will protect the DNA. So it's like grips the DNA tightly. And only upon phosphorylation of ABCDE, it will let the DNA go. So the DNA end can be repaired and stitched together. If you don't have its phosphorylation happen on this site, then the DNA never gets repaired and then become a problem for cell if you have a broken DNA end. So phosphorylation is to let go of the DNA. So if we want to see how it's phosphorylated, but phosphorylation will make it release DNA, it's not a very good way to look at the structure. But the nice thing is that, so we know that the DNA PK is required for activating the Artemis. So Len, a very talented postdoc, she was looking at the time course of autophosphorylation, and you can see it's phosphorylating itself, also phosphorylating the Artemis. There are a lot of phosphorylation site, and the reaction is pretty much complete within 10 minutes. But the nucleus cleavage of the hairpin by Artemis is a very slow reaction. So when the kinase fully phosphorylates itself, there is only like a very small percentage of nucleus cleave the DNA. So this time difference gives us a chance to look at the phosphorylated, activated Artemis, but DNA hasn't been cleaved. And that's exactly what happened. So before it's autophosphorylation, we already see the ABCDE cluster. It's a large, like 200 residue region, was hanging out outside of the kinase region. It's not phosphorylated. And once we have the hairpin, DNA substrate, and the Artemis, then it's phosphorylated. We saw 4 different sites get phosphorylated in this cluster, and it's become very well structured. And the structure is not like alpha strand or alpha helix or beta strand, but it's formed this like a grip. We call it like almost like a jar, biting to the green ring, the top of the green heat repeat. And we'll see a little more of this. So this is not the best coloring, but those are 4 phosphorylation sites with the peptide around, wrapping around this top of the green ring and biting to it. If we, as I told you, if we mutate the kinase active site or mutating the phosphorylation site, it's all dead. It cannot be doing the NHEJ. We collaborate with Cassie Meek, who is in Lansing, Michigan. So they also mutate the sites that recognize phosphorylate threonine. So what happens is that you can either mutate the hydrophobic residues or positively charged lysine arginine, mutating them also eliminate BDJ recombination. So hydrophobic or positive charge. So it's very clear to phosphorylate itself and binding to the heat repeat is very important. And the conformational change I told you will release the DNA, we visualized it. So basically that's looking at the bottom of the DNA PKCS, that's where DNA is bound. When it's become phosphorylated, the groove opens up from 25 angstrom to 50 angstrom. So the DNA without any help will be released. And so DNA end can be processed, right? So our case, we have this Artemis there. So now we can see the side view of this autophosphorylation binding here, change the conformation of the side view. So you can see the big opening between blue and the green heat rings, and Artemis inserts itself in between. An active site can grip onto the hairpin to cleave. So there's this requirement of DNA PK to help the Artemis to bind. Artemis have the catalytic domain here, and then there's a long neck, like a swan neck, hanging onto the top of the PKCS domain to make those interactions. And I showed you the structure earlier. By itself, it can only bind a few base pair of double-strand DNA with 5' end in the active site. And Artemis is actually remodeled by DNA PKCS. This is a pink loop by itself as a loop, but with the presence of DNA PKCS, it forms a short helix. And by restructuring, it becomes able to bind hairpin end. And so this new structure is stabilized by DNA PKCS. And not only the active site is remodeled, also Artemis only can bind a few base pair near the end, near the hairpin end. The rest of the DNA all depend on the DNA PKCS showing blue and the green or cool to stabilize. So you need to hold the DNA so that the hairpin tip can be cleaved by the Artemis. So with the remaining 5 minutes or so, I will give you a quick run over through the NHEJ. So each structure, the beginning of the regular record 2 took us 20 years tempting and 10 years to get structure and 2 or 3 years eventually to have every step. And then with cryo-EM, it just speed up. And NHEJ, once we have the methodology, it was just like 1 year, 2 year things. So Len was a postdoc I mentioned. She did the Artemis and DNA PKCS, which is a very tough project. And then she had 2 babies during the process. First baby was born after Artemis structure was determined. She just had a second baby. So I think science not necessarily exclusion of like having family. So she actually worked out the NHEJ, which is join the open end. And not only join the open end, like here, the blunt end easy to join. And there's also polymerase involved. There's a special class of polymerase that's being recruited to insert more nucleotide so that it can generate more variation when it forms the antibody antigen codon. So I'm not going to give you a lot of detail, just give you a glimpse of what she did. So she generated the DNA substrate. So because we want to make endless of DNA, so it's only one end of DNA is workable. So this is copying Jonathan Goldberg's work, just making this T junction. So it's sort of the DNA PK wouldn't bind. So it's kind of protected. Everything's focused here. So if we have either a nick, so ligase can ligating, or a gap, so polymerase can insert more nucleotide, and opposite another strand, which is no action can be taken because we have the 3' OH, 5' also OH, so no ligation. Sometimes we use 3' dideoxy, so we only focus on one strand. And so that's a substrate for non-homologous end joining, minimum base pair or even no base pair between the 2 left and right DNA. And it was known all the players, each player structure was determined. So you already saw the Kusamani bind to DNA. There's a special family called X family polymerase. We're using here just Tomu as an example. Tomu can put 2 DNA ends together without any base pair. It can use the opposite DNA end as a template to insert a new one. So it's a pretty cool enzyme. And then the ligase 4 is dedicated for this non-homologous end joining, and structure of ligase binding to a DNA nick was also determined. And besides those essential proteins, there's also fillers, which is protein somehow to assemble the two DNA end together. So there's XRCC4. Ligase have two C-terminal BRCT domain that binds to XRCC4. And they together called L4X4 is the scaffold somehow to put the two DNA ends together. Structure of that was determined a long time ago. And then there are two more things identified in 2007, 2015. XLF is XRCC4-like factors, and it's somewhat related to that, have the head dimerization domain and then alpha helix extension. And the last player is called the PAX, and also have the same dimerization, but doesn't have a long helix extension. And those proteins have the wiggling C-terminal peptide will bind to Ku. So those are all the players. And Len just made those protein, put it together with a bit of finis to refine the protein and the DNA to get really high resolution structures like the complex. So I will just show you how the scaffold come together. So we call it omega framework. So you have XLF in the middle, the dimer, XRCC4 on the dimer on both ends. So it's form a very large omega framework, like 150 angstrom here, 240 angstrom on top, and extension is about 120 angstrom. So it's a big framework. And with ligase added BRCT domain, you can see everything will happen in between these two. And the DNA comes in, two different DNA. It looks very nice, suspended on top, like a trapezoidal structure. But if we turn 90 degrees, there's no protein DNA interaction right now here. So the scaffold and the DNA is linked by Ku70, Ku80. Ku70, Ku80, as you've seen before, binding not exactly at the end of the DNA, but like one helical turn inside. And that interaction between Ku70, Ku80 and the BRCT domain of the ligase dock the whole DNA onto it. And this trapezoidal framework is very nice because it has space for enzyme to work. It's nicely keeping things together, but it's not rigid. It's the 4-point things, so we can do all those wigglings. And then eventually, so this is the DNA end to need to be put together. So we have two different breaks. This is the AMP we put here, just marking where the ligase can work. So ligase and the polymerase will access the DNA here. All the nick and the gap have to face outside where the enzyme active site will come in. And there will be a toggling between. So the cool thing is that if the ligase and the polymerase is attached to this side, they only work on one strand with one polarity. And the other side work on the other polarity. So two ligase, two polymerase, and they have specificity, and they have to switch and toggle to see if it's ligatable, it's a ligation. If not ligatable, it's polymerase. So it's really cool. There's 4 enzyme toggles back and forward. So, and I think it's pretty complex, and there's things to help the two end to put together. That's the PAX, which have C-terminal peptide will bind to the Ku70, and then have the dimerization domain was swimming there. We just couldn't find it. But the important thing is to reinforce two DNA ends are linked. So yeah, so ligase coming to ligate to the top. And the only thing we find that novel, because each part is known, we know the framework, it's that the ligase never goes away. So when polymerase come in, two domain of ligase will be floating out, but there's one domain of ligase holding DNA together to help the polymerase to get access to the DNA gaps. So this is summary of the NHEJ framework for ligation and everything you have seen already. And then for the polymerase, and as I mentioned, everything is the same, except the DNA bending angle is much more sharper than the ligation. So there is toggling and there's a switch conformation. And when polymerase work on DNA gap, the ligase still donate the domain to help it to keep it together. So, and that's it. And I think I'm concluding my talk here that, so what we learned is a lot of detail, but on the large sense that every nucleus in cell is regulated because you don't want the nucleus going anywhere to be active. So it need a very, very tight regulation at the protein expression level, protein partner, and the substrate access. You can have activator, you have inhibitors. And then the non-homologous end joining, it's depend on the many factors put together a framework, and there's stochastic toggling, but there's also cooperativity. One help the other to put things together. This is actually a pretty cool movie that how the ligase and the polymerase switch place. So the ligase, when it's active, it encircles the DNA and accesses a nick here. But when the polymerase is there, the ligase is giving its way, but the polymerase and the ligase have toggling, and they all attach to the right side for the access one strand. So structure biology is cool. And even though the technology is similar, you're using similar tools and approaches, some similar, but we always discover how new things happens, the mechanism of how macromolecule assemble and regulate and achieve a particular purpose. So the work is done in a long-term collaboration with Marty Gallett. I was interviewed at NIH in 1995. He proposed, because Marty's lab reconstitute the first BDJ recombination. He said, if I join NIH, we can collaborate. It was very generous of him, because some scientists feel like, that's their project. You have to apply to get to work on that. So I joined, and we collaborate. And the first really major structure was solved is almost 20 years after I joined NIH. And our collaboration is still going on. Marty is 94 years old, and that's the beauty of NIH. And he still is very actively involved. And the project took many years, so I credit people along the way, and currently Len's the one working on the project. And we have collaborator, and that's funding source of NIH. Thank you.
Questions for Wei?
So I had a question about Artemis, and it had what you call the swan neck.
Yeah.
And I was wondering if that region of the protein is essential, and if you think Artemis could always be there, kind of tethered by that domain, and then just swing in when it's needed.
Yes, it's an accessory, and people but show that you need that 400 residue, including this bit of swan neck, to have it efficiently being recruited. So it's probably hanging out there, waiting to get in. And that binding site is also a recruitment site for XSCC4. So it's like there's a lot of toggling, competition, cooperation. Yeah.
So I have a question about the, hi.
Yes.
Yeah, I have a question just about the hairpin. So is the sequence in the hairpin I mean, is it normally the same, or is it always different?
It's not always the same, because it's coding region of the V or D or J. So it can be any sequence, but there are preferred sequence happen more often.
Are they often like CT rich?
It's more AT rich.
It's more AT rich. So is it then, can it be targeted by ApoBex? Do you know?
It's possible, right? Yeah. So ApoBex play a very important role in the next step in class switching. But hairpin and
I was just thinking if there were Cs there.
There are Cs there sometimes.
Yeah,
but,
it might be.
Yeah, it could.
And then just something interesting, Chris and I did our undergrad together, and you're just lying there, Chris Buell and I did our undergrad together in the biochemistry department. I didn't know you were working with him, so.
Yeah, we cannot advertise that, because for years and years, nothing happened.
Any other questions?
Yeah, it's a lot of material, but it's an overview. You need to remember all the detail.
So Wei, I have a question regarding the last movie that you had on the last slide. So I saw that there's it's dimer. There's two cool dimers on both sides, but only one is turning.
Right.
So maybe can you comment a little bit why one is turning and one is relative to the other?
Very good eyes. So when we compare the structure, because when the polymerase and the ligase attach on one side, they work on one strand. So we only focus on one. The other side are all floating. So we call this side as active site because things happens, enzymes there. The other is supportive site. So we have polymerase structure, the one assembly, ligase, another assembly. When we compare the structure, we find the supportive site just doesn't change very much. You can superimpose any part. They do very small adjustments. It's the active site that do big movement. So supportive site somehow doing supporting structure.
So is it sequence dependent, or because there's a gap, 1 base pair gap, and then there's a 0 to 3 base pair
Right.
gap. So there's a asymmetric way.
Right.
It depends on that structure.
It could be because we haven't looked at the other structures, but we figure like if the substrate is same, except if the gap, the other side, then now working one is a gap, same size, then the supportive site will always have the same configuration. They have to rotate the DNA into the right position for polymerase or ligase to access.
Right.
So I have to expose the outside.
Right. Any more questions for Wei? If not, let's give her a round of applause for this fabulous talk.
Thank you.